today I'd like to show you some work we've been doing over the last years, for some years now, together with the Bibip team, and where basically we combine microscopy and mineralogy approaches with biology approach, especially bioinformatics. And I'd like to show you how this combination works very specifically on one topic that we have been interested in, which is the formation of minerals of amorphous calcium carbonate by bacteria. And I'll finish by that. But basically, most of what I'll show you is based on a paper that was just published in Genome Biology and Evolution, and I'll tell you why we got to that journal and how and so on. I mean, that will be for the end. So there's a bunch of authors of this paper, and you'll see there are many more collaborators on the work that I'll present. And among those authors here, I mean, you have a really significant amount of people from IAPSC, but also from the I2BC at CEA Saclay, and the Ecology, Systematic and Evolution Research Unit in Université Française. Okay, so yeah, I think the story starts there. It starts in 2010. And at that time, we were working on stromatolites, the formation of those carbonate rocks in lakes in Mexico. And we were going to that field site, which is called Lake Chichica in Mexico. And what you see, I mean, you see the walls of the volcanic crater here, and the center is occupied by this lake. And what you see here, all these rocks, I mean, they are whitish when they are out of water, and they are greenish when they are beneath the water surface. And all these rocks are the stromatolites. So these are carbonate rocks formed by microbes. And so we took pieces of these rocks, got them back to Paris, and these were basically the colleagues from Orsay. Put that in their aquaria, but at some point, if you want to visit also, we have also aquaria like that. So we put these pieces of rocks colonized by microbes, put them in aquaria, and let them live for some time. And I mean, and that was really, I mean, I was talking about serendipity in the abstract. This is how it went. I mean, at some point we say, oh, let's have a look at how this looks like. Just use the microscope, scanning electron microscope, and let's look at how the microbes look like, and do we see some association with minerals? So we took some drops of that of this aquarium and looked at them by microscopy, and this is what we discovered. Of course, there are many different diverse microbes living in those aquaria. But among those diverse microbes, we were struck by this one. Here you see a false color scanning electron microscope image of a cell that is deposited on a filter. So the blue stuff is the filter with 0.2 micron pores. And so you see a cell, and this cell is filled with those globules that we analyzed by diverse techniques and showed that they were amorphous carbonates. And so we also identified this microbe, and this microbe is a bacterium, and more specifically, it's called the cyanobacterium. And what are cyanobacteria? So they are very diverse bacteria. They form a single phylum, and they perform this metabolism that is called the oxygenic photosynthesis. And basically, so they have pigments like chlorophyll, other pigments as well, like cyanine. So like plants, they use CO2, H2O, and using light, they can oxidize water, take an electron from water, put it on CO2, and so produce reduced carbon, so this is organic matter, and release dioxygen. And so you see here some light microscopy images of these very diverse cyanobacteria. They are all cyanobacteria, diverse morphologically, of course, but they are all doing oxygenic photosynthesis. And of course, for geologists, this reaction has been really pivotal in the history of life, since with the advent of oxygenic photosynthesis plus other processes, at some point we got oxygenation of the atmosphere at the surface of the Earth. So this massive production of oxygen, of dioxygen, that eventually built up in the Earth's atmosphere was invented by bacteria. It was invented by cyanobacteria. It's only later on that some eukaryotes, some algae, also acquired this capability to perform oxygenic photosynthesis. You know that plants or algae can do that as well, but this was due to some symbiosis with cyanobacteria. So again, production of dioxygen is an invention that appeared in bacteria a while ago, so most likely before 2.3 or 2.4 billion years ago, since this is the time when we start seeing the dioxygen building up in the Earth's atmosphere. Another interest in for these bacteria, these cyanobacteria, is this. They have been studied for a long time in neuroscience because we think that they are involved in the formation of massive carbonate deposits. And the way we explain this is the following. So here you have a chemical reaction of the calcium carbonate precipitation. So in any aqueous environment, you may have calcium 2+, you may have bicarbonates. And so if you have enough of those two things, of those two ions, they may combine, precipitate calcium carbonate and release CO2. And so, as you remember, cyanobacteria are performing oxygenic photosynthesis, which means that they sequester, they trap CO2. And so since they trap CO2, they tend to displace that equilibrium toward the right, hence the precipitation of calcium carbonate. So cyanobacteria, because they are doing oxygenic photosynthesis, they tend to favor the precipitation of calcium carbonate. And what we know is that eventually they can form up these rocks that are called stromatolites that I was referring to in the beginning in Mexico. This is one section of a stromatolite from Sardinia that is studied by Bibip. And we know modern stromatolites, but we know also ancient stromatolites up to 3.5 billion years ago. And they are among the oldest traces of life on Earth. And this is really based on that idea that microbes favor the precipitation of calcium carbonate. And for a long time, we thought that cyanobacteria were mostly responsible for the formation of these rocks. And the way we used to think about the formation of calcium carbonate by cyanobacteria was the following. And I mean, I already explained to you that basically, because they are performing oxygenic photosynthesis, they are sequestering CO2, and therefore they displace the chemical reaction of toward the calcium carbonate precipitation. In a little more detail, you have this sketch here. So basically what we believe is that they take up bicarbonate ions from the environment. They spend energy to take a lot of these bicarbonates within the cells. And then within the cells, there is a compartment here, which is in red, that is called a carboxysome. It's delimited by proteins. And this is where basically the reduction of CO2 in the reduced carbon, in organic carbon, takes place. And so you see that at some point you have transformation of bicarbonate to CO2, and that releases OH minus. And the idea so far was that this OH minus somehow get kicked out of the cells. So you have an increase of the pH outside of the cells, and that induces the precipitation of calcium carbonate. So for a long time, people had thought that this calcium carbonate formation by cyanobacteria was only extracellular. And there were like several outcomes from that paradigm or that idea, is that basically the chemical composition of the carbonates that you form outside of the cells depends on the chemical composition of the solution outside of the cells. So if you are in a lake with a lot of magnesium, you may form magnesium carbonate. If you are in a lake with a lot of strontium, you may form strontium-rich carbonate. Now, with the idea that, I mean, with the observation of some cyanobacteria forming intracellular carbonates, we completely changed that idea. I mean, the chemical composition of the carbonates they form depends on the chemical composition inside the cells, which of course is quite different from outside of the cells. And I'll show you some examples. And the other idea is that with this extracellular formation, people used to think that, okay, cyanobacteria somehow form calcium carbonate very incidentally. I mean, this is some kind of secondary reaction, secondary byproduct. This is not intended by the bacteria. And in the case of intracellular carbonates, you may imagine that you need some specific mechanism so that you will not precipitate calcium carbonate inside a cell in any way at any cost and so on. I mean, this has to be regulated by the cells. So there are probably specific genes that are involved in the formation of intracellular carbonates. So again, past view was like, okay, they don't want to precipitate carbonates. New view with the intracellular ones, maybe they really intend to precipitate this carbonate and there might be a function. But I'll get back to this. Okay, so as I told you, so about the implication of the geochemical implication, first of all, what we observed culturing these cyanobacteria forming the intracellular carbonate is that some of them, they can form calcium carbonate in solutions which are undersaturated with calcium carbonate, which means that basically, if you take the solution in which they live, these cyanobacteria live, thermodynamically, you can show that you should not form any calcium carbonate. But within the cells, those bacteria can. And of course, there's an energy expense that explains that basically they build up oversaturation within the cells. And basically what it means is that you can observe formation of carbonate in an environment where you should not have calcium carbonate. So that's first thing. The second thing is we observe also some unusual element fractionation by those bacteria and those carbonates formed by those bacteria. Here, what you see is that, so we cultured cyanobacteria in a solution where we added barium, strontium, and calcium. And here we are looking at the evolution with time of the concentration of barium, strontium, and calcium in the solution. So you see that in the first step, you have a decrease of the barium concentration in the solution, which means that basically the bacteria are sequestering barium, and the concentration is decreasing in the solution. And once they have basically sequestered all barium, uptake all barium, you see that in red you have the strontium, then the strontium sequestration starts, and when there's no strontium left in the solution, then the calcium starts. Which means that, and we could observe by electron microscopy that we have like those layered carbonate inclusions with layers of barium carbonate, then strontium carbonate, then calcium carbonate. So that means that those bacteria, when they look at those different elements, so all these elements, they are alkaline earth elements. They are in the same column of the Mendeleev classification table. And basically, they have relatively similar chemical behaviors. Those bacteria, they are able to really efficiently discriminate those different alkaline earth elements, which is pretty unusual. If you precipitate a carbonate under abiotic conditions, basically the amount of calcium, strontium, and barium you have in the carbonate is about the same as what you have in the solution. Here, you may have a lot of barium, even if you have very little barium in the solution. Another striking thing was to realize that those carbonates inside the cells, they are amorphous. And amorphous carbonate, amorphous calcium carbonate, they are usually, again, under abiotic conditions, they are usually very unstable. They tend to transform spontaneously into the more stable and crystalline compounds such as calcite or aragonite. But in that case, every time we look at those bacteria, they are amorphous. So there's some way, there's some process that makes stabilization of the amorphous carbonate. So that was another surprising thing. Last, and that's related with basically there's an unusual fractionation pattern that I told you about. It seems so they prefer heavy alkaline earth elements. They take heavy alkaline earth element preferentially to the light ones. So basically you have magnesium, calcium, strontium, barium, and radium. And what we showed is that if you start feeding them or put them in a solution where you have some radioactive isotopes, I mean, radium has only radioactive isotopes. You can also do that with a radioactive isotope of strontium, like strontium-90. Even if you have tons of calcium in the solution, they will take very efficiently those radioactive isotopes at very low concentrations. And this is what, for example, Mehta here showed with using nanosims, that basically, even at very low concentration of radium mixed with a lot of calcium, the radium isotopes are taken up by the bacteria and put in the carbonates. So that's very interesting if you think about depolluting some environment where you have calcium, which is the case in any environment, and very little amounts of pollutants. But yeah. Okay, so in the end of all these things, our first questions, or at least our question at some points, were how many cyanobacteria can form intracellular calcium carbonate? So far, I've shown you one, and that one we discovered it by chance, just by microscopy. Okay, and so the first question we got was, the first comment was, oh, this cyanobacterium is nice, but okay, that's is that like an alien and something very, very unique, or is this the process that could be more widespread and people have overlooked it? What makes those cyanobacteria different from the ones that are not forming intracellular carbonates, and how abundant are they in the environment? The second question as a geologist was like, okay, since when this happened? I mean, is that something like is recent at the geological scale, or is this something that is ancient, this formation of intracellular carbonates? Because of course, thinking about stromatolites several billion years ago, were cyanobacteria at that time forming more intracellular carbonates or extracellular carbonates? And last, what are the mechanisms of this biomineralization? Of course, if you want to better understand how to stabilize amorphous compounds, or if we want to know how to fractionate different alkaline earth elements, we need to learn about the mechanisms that are used by those bacteria. So, of course, to answer these questions, we've done a lot of experiments, sometimes pretty time and energy consuming. We used cryo-electron microscopy like Cenovis to look at these carbonates. Here you see the globules of carbonates inside the cells. We needed cryo because if we use more standard electron microscopy or more standard sample preparation, those carbonates, they are just gone. I mean, we lose them. So we needed cryo. That was painful, but it worked pretty well, and we could observe some envelope around those carbonates. So they are formed within a compartment within the bacteria. We did also some cultures in the lab, and so we could observe the variation of upon day and night. So during the day, of course, the cyanobacteria, they are doing the oxygenic photosynthesis. During the night, they are not. They are respiring on oxygen. And so this goes with variations of pH of the solution. And we could observe that calcium is sequestered all the time, even during the night. But okay, so and so on and so on. But you know, despite these intensive efforts, we got the answers. That was nice, but not really answers to the question I just asked. How many of these cyanobacteria? What are the mechanisms? We are progressing, but that's slow. And this is why we started setting up this, let's say, bioinformatics-mineralogy approach together with first Isabelle and Elo. So getting back to it, we start from that cyanobacterium. Okay, that was discovered, as I told you, in 2010, published in 2012, just to have some time reference. 2014, so what we did is that in order to answer this question, is that the only cyanobacterium doing it, or are there other cyanobacteria forming intracellular carbonates? We went to a culture collection at the Pasteur Institute, and over there they have hundreds, thousands of strains of cyanobacteria that they are culturing. And so we asked for some strains that, some 1 milliliter of their culture, so that we could bring it back here and look at them by scanning electron microscopy. We observed 70 of them. So this was several days of microscopy. We tried to work early in the morning or late in the afternoon. And so we discovered this way that other cyanobacteria were forming intracellular carbonates. And these are the names they have. I miss here the usual name, like the genus and the species, but PCC for the Pasteur cyanobacteria collection. The first 2 digits are for the year they have been found or isolated. So 1972 for this one. And the last digits are for the number during the year. So this is the 3rd strain that was isolated in 1972. So you see that this collection has been maintained for some time now. It's more than 60 years, and they have it's very, very useful. So we found 7 more cyanobacteria forming intracellular carbonates with this approach. So that makes now 8 cyanobacteria forming intracellular carbonates. Interesting thing is that for 58 of these strains that we have been looking at, we add the genomes. Okay, so let me pause here just to get back to the basics of in biology. I mean, that's basically the level I have, so that's good. I can share that with you. Just to remind you, okay, in a bacterium, there is a genetic material, which is and there's this genetic information that is carried by this molecule, which is the deoxyribonucleotide acid. Otherwise, it's called DNA. And this molecule consists basically is a polymer, and it consists of the alternation of different nucleotides. And we have 4 different ones, which are thymine, adenine, guanine, cytosine. So and they are basically abbreviated by A, T, C, G. With those 4 letters that you can, sequence in any way, you have the information. You have some information, and so this you'll see that this is one way to represent the whole genome of this bacterium, so the whole chromosome of this bacterium, and all these lines represent one gene. So just for the order of magnitudes, one genome in one bacterium is usually about few millions of nucleotides, few millions, and that means basically, that also means a few thousands of genes. One gene order of magnitude is 1,000 nucleotides. For cyanobacteria, the size of the genomes varies. That basically it can be up to 16 million of nucleotides. It can be down to a million. Okay, so that's for the genomes. Information. How does it work? You have DNA here with the ATCG sequences. Some of the genes, they are transcribed in another molecule, which is called RNA. And this messenger RNA can be translated into a polypeptide, so sequences of amino acids. So it's another type of polymers with different monomers. Some other genes, they won't be translated into proteins. They will transcribe in other RNA that have some functions in the cells. Sequences. So you have the sequences here of the genes, A, T, G, A, T, C, and so on. When you transcribe that into the RNA, basically you have the same letters, except you see the T is replaced by the U, whatever. This is then translated into proteins, and you see that 3 nucleotides here, which form what we call a codon, gives 1 amino acid. So they correspond to 1 amino acid. And that's what we call the genetic code. So there's a code which tells you if you have an AUG codon, you get here this methionine amino acid. Okay. What you have to remember is that those here we are interested by those polypeptides. When they are long enough, we call them proteins. Protein, they can have different roles in a cell, but basically they are really the tools, the actor of all the biological functions of the cells. So getting back to our formation of carbonate, this is to these guys that we want to look at in order to understand who is doing the carbonate. Okay, one last step. Having the sequence, the amino acid sequence in proteins is one thing. This is very interesting, and all the information is there. But actually, those polypeptides, they are not just linear polymers like this, okay? They tend to fold. And this is because of this folding that they start acquiring their functions and their chemical properties. And then you can define what we call the functional sites, which are usually located within the proteins. Some location. You will define their surface properties and so on and so on. One difficulty is to, so as you see, having the sequence of the proteins, the linear sequence of the protein, we can get that from the sequence, the linear sequence of the genomes, of the genes. But going from that linear sequence of amino acids to the structure is tricky. That's not trivial. And basically, predicting these protein structures and function is really not trivial. And this has fostered a lot of work in structural biology. And I think, maybe I'm wrong, but I guess this is one big work that has been done over many years here at IMPMC by people working in structural biology, trying to understand how we get from there to there, and then understanding the function of these proteins. Okay, so that's it for the basics in biology. Let's get back to our story and our questions. As I told you, we have 58 genomes of cyanobacteria, and some of them, we saw 8 of them, they are forming intracellular carbonates, and the rest are not forming intracellular carbonates. So here you see you have 58 genomes, and the ones that are circled in red are forming intracellular carbonates, and the other one are not forming intracellular carbonates. And so the first question was, okay, are there genes which we find only in those red squares and not in the other genomes? So are there genes that we only find in cyanobacteria forming carbonates and that we don't find in cyanobacteria not forming intracellular carbonates? You remember, each of these 58 stuff is like a million nucleotides. So you have to deal with this huge load of information. And so results of this study was like, there was one single gene that was shared by all the 8 cyanobacteria forming intracellular carbonates, and that was absent from the other 50 cyanobacteria not forming intracellular carbonates. One gene. We'll discuss that later, but and basically this is the way we see it. So this is, we align, so not the nucleotide, but the amino acid sequence of this protein for different cyanobacteria forming intracellular carbonate. And we see 2 things. We see that they align pretty well. I mean, we can see the same amino acids at the same positions in these different proteins, in this protein for these different cyanobacteria. Sometimes we see some differences. And basically these differences are due to the evolution. those bacteria acquired this gene, and then there was some evolutionary mutation, and so sometimes you have some differences between the sequences in these different bacteria. Okay, so we have one gene. And so what we did is that what we can do then is, your question is, what is this gene? What is the function of this? The nice thing with this is that you can try to get an answer directly by going to public databases. And for example, these public databases from the NCBI, there are several hundreds of millions of sequences that are recorded in that database of proteins, and you can compare your protein sequence to all the protein sequences that are or that are present in the database. And so this is exactly what you do. So you have your sequence or your sequences, your 8 sequences, and you compare them with the database and try to align them, to align your 8 sequences with the one that are present in the NCBI database. And you get a result. And this is not the result we got, but this is just an example. You get a result here. So if they align over some lengths, I mean, you are really happy, because that means that you found in the databases, you found sequences that are matching your sequence, and so they might be the same proteins. If they match only over a restricted area, okay, you are maybe a bit happy, but not so much. And of course, if they don't match it at all, okay. And so the result for ours is that there was nothing matching. Okay? There is no detectable, at least full-length homologs with the sequences that were known in NCBI. So you have mixed feeling with this. You can be either quite depressed because you have a gene, but you have no clue what it is and what it is for, so you don't have your answer. You can have your excitement because you feel like, oh, this is still new, and so we have to go on and go on. So this is what we did. But basically what we know from there is that we have a gene that we call CCYA and a protein which corresponds to a protein, okay, that we call calcyanine. And this is a new gene, new protein. And we don't know the function. But what we know is that this gene is a diagnostic marker of cyanobacteria forming intracellular carbonates. And so that was the interesting thing because, before going there, so we have something unknown, okay? We don't know that protein, we don't know that gene. And yet, there are some magicians who can tell something about unknown things using some approach, here the hydrophobic cluster analysis, where, and I won't dwell into that, here what you see basically is some way to represent the amino acid sequence of this protein, so the calcyanine protein. And you have a color code with different different amino acids with different chemical properties or different physical properties. And for example, you have those green amino acids here, which are circled or outlined by these black lines. They are hydrophobic amino acids. And those hydrophobic amino acids, what we know is they tend to cluster and they tend to drive the folding of the proteins. So they will be very good marker of the structural motifs in a protein. And so by looking at those, this kind of graph, it was possible to see several things. First of all, there are some structural motifs here, like this one, which is repeated a second time and a third time. So that old stuff is a triplication of one single motif with some variation, of course, but basically they look relatively similar. And this motif looks some known motifs that we find in other proteins, and they are called the glycine zipper motifs. So we find them in other proteins, but in that case, there are also some differences. So in that case, in the calcyanine, these glycine zipper are longer than the one we usually find in other proteins. So that's the first thing. Then, so you can separate that what we call a domain, which is an element that is stable and folds independently of the rest of the protein, from another domain, which means that calcyanine basically is composed of two domains. There are two domains, so two parts which might fold independently. And so what we could see also is that this domain here, which we call the C-terminal domain, is pretty conserved. It's very close from one cyanobacterium to another. And this one is very variable. There's a lot of sequence variation in that domain between the different cyanobacteria. Okay, so back to the previous slide. So we have a gene or a protein that seems to be diagnostic, that is a marker of the formation of intracellular carbonate by cyanobacteria. So now what we can do is use the sequence of that gene or that protein and look in the databases whether we can also find that in other cyanobacteria. So we have, for example, we can start with our the sequences of the 8 different calcyanine from the 8 cyanobacteria we found in the Pasteur collection. From that, okay, we can identify some of the sequences which provide this probabilistic signature. And then we confront those sequences to all the genomes that are available in the databases, genomes of cyanobacteria or other bacteria. And from that, we may find or fish new sequences of calcyanine, add them to that multiple alignment, and so on, and then we do it again. So this is an iterative process. And of course, we have to do that from time to time, since the genomes from NCBI, they will, every week, they grow and larger. So doing this, in 20, so we did that the first, one of the first time we did that in December 2017. At that time, we had, sorry for that, I can't probably, during that time, in the databases, we had 600 genomes that were of cyanobacteria that were available in the NCBI database, or in the databases, in the public databases. So we searched into these 600 genomes for calcyanine. We found 27 new strains. So, that was, for me, that was really a gain of time, of course, because I didn't have to do the electron, yeah, I did some electron microscopy, but this is was really some work for people doing the bioinformatics. But yeah, so that was impressive. Okay, that was impossible to scan or to screen 600 different bacteria by electron microscopy. So at that time, so we confirmed, we checked back several of them by electron microscopy, getting these strains from different collections around the world. And we could confirm that they were containing intracellular calcium carbonate. And that was really nice, because we also, so those cyanobacteria, they come from everywhere around the world. Some of them we discovered they are multicellular cyanobacteria. So a bacterium usually, it's a single cell, but in cyanobacteria, some of them, they really attach to each other and they form filaments. And sometimes even those filaments are branching. So it's pretty complex. And so we found some of this cyanobacteria forming intracellular carbonates. We found also some of them in some cyanobacteria which have received a lot of attention, and I'll show you that in this genus called Microcystis. This is a sometimes toxic cyanobacterium, which is blooming in some aqueous environment. And I'll get back to that, but that was pretty interesting. And we also found the calcyanine in some marine cyanobacteria. So far, we had only freshwater ones. So you see, it expands really the environmental important significance of this cyanobacteria. Yeah. So now we have 27 plus 8, so 35 cyanobacteria forming intracellular carbonates. the question is, how are they related to each other phylogenetically? So to do that, I mean, again, using the genomes, you have some information about the phylogeny, so the genealogy somehow of these cyanobacteria. So you can compare some genes which are shared by all these cyanobacteria, and you compare those genes, and if the genes are very close in sequence to each other, that means that the bacteria that are borrowing those genes are very close phylogenetically. Now, if you have a lot of mutation in the sequences of the same gene, that means that they have diverged very in the past. Okay, they have diverged significantly. So what you see here is some kind of genealogical tree. Each line is a cyanobacterium, and the closest they are in the tree, the more related to each other they are, phylogenetically close, they are close cousins somehow. In colors, what you see in colors, all the colored ones are the ones with the calcyanine. So they are forming the intracellular carbonate. And what you can see is that you have colored ones everywhere, you know, a different place of this tree, which is representing most of the diversity, the non-diversity of cyanobacteria, which means that basically we find cyanobacteria from intracellular carbonate pretty everywhere in the tree. So there are two possibilities there to explain that pattern. The first possibility is that this calcyanine appeared very early in the history of the cyanobacteria, at least there. So, and it was transferred to all the descendants, but lost by many lineages. So all the non-colored, basically at some point of their evolution, they lost the gene encoding for calcyanine. So that's one possibility. And I mean, this is usually the way we would interpret. I mean, if we were looking at genealogy in humans, for example, this is the way we would interpret that. There is an alternative though. The alternative is that the gene appeared more recently by some of these organisms, and then it was transferred to the other bacteria. And this is what we call an horizontal transfer. These bacteria, they are able somehow to exchange DNA between each other, you know, when they are living in the same environment. They don't have to be the same family. They can exchange DNA. So that's an alternative. So in order to discriminate between these two options, what we did is that we looked at the phylogeny. This time we compare all the calcyanine sequences. And we look at the phylogeny of the calcyanine of the CCYA gene. And this is this tree. So this is based on the sequences of the CCYA gene. And in parallel, we look at the species tree. So we don't look at the calcyanine gene or the CCYA gene. We look at all the other genes which tell us about the phylogenetic relationship between the bacteria. And when we compare those two trees, they are very congruent. They are the same. I mean, they branch, all the bacteria branch at the same level. And this is indicative of ancestry of the gene. So that means the preferred scenario is that this calcyanine appeared very early in the history of cyanobacteria. It was transferred to all the descendants, but lost by some lineages. When did it happen? We don't know. This is super debated, and some, yeah, okay. There are regularly some debates, but I just picked up one study. Okay, there are many others giving different numbers, but in these studies, for example, so they turn this tree this way to this way, and in this way you have time this time. So that scale for them is time. So basically, they look at how many differences in a given gene between two sequences. And the more you have, the longer, the higher the time. Okay, so basically they look at those different divergence and put them in a time scale. And so these are the different cyanobacteria, different cyanobacteria. Here, based on this, I mean, we can infer that our calcyanine would be located at least here. And so if that time scale was accurate, you see that puts us up to more than 2 billion years ago. So this is a node. This is a node gene. Okay. So what is the role of calcyanine? And you see now it's going to be quick because we don't know much. But again, still based on this information that is available in the databases, we can infer several things. First of all, I told you that this calcyanine protein is composed of 2 domains. And I told you that one domain, which is the one that is on the N-terminal, domain is pretty variable. In some cyanobacteria, this domain looks very similar to domains that are known and which are called high metal affinity, heavy metal affinity domains. So in our calcyanine, we have a domain that looks close, not the same, but close to domains that are known for their affinity for heavy metals. So, of course, the big question is how this calcyanine interact with metals and cations. The other interesting thing is that we can look at the genome. So the genomes is not, what you can look is how the genes are assembled within the genome. And here, for example, you have one sequence of a genome of this cyanobacterium, which carries calcyanine. And the gene is here. And when you look at the genes that are close by, upstream or downstream, it's interesting to see that, the one that is right next to it is a calcium proton exchanger. So transporters of calcium. And of course, if you want to make a calcium carbonate, that's good to have. And there is also here, there's a carbonic anhydrase, which is that enzyme that converts the bicarbonate into CO2. So that's awesome. And you have also many gray stuff close by. You could tell me, yeah, but who are they? And basically, we don't know. Several of them. So this gene or this protein is most likely involved in the formation of carbonates, intracellular carbonate. We don't know how, but it's involved, and for sure, it's a marker of this biomineralization. Okay, so perspective now, or where we are right now. First of all, as I was telling you, there is a really dramatic increase of the information in the databases. I told you about this detection of calcyanine or CCYA in 2017. We had 600 genomes. Last March, we had 3,000 genomes of cyanobacteria. And now we have I don't know what is the latest number, Maxime, maybe you can tell me, but right now my latest number is 164 strains with calcium. It's a bit more, probably, but okay. So Maxime has set up that tool where basically, so the tool is doing many things, but among these many things, you can really look at search for this calcyanine or the CCYA gene in a updated list of genomes that get available. So right now, you see that number. I mean, if you take a rough ratio of that, the strains cyanobacteria with the calcyanine, they are only 5% of the total cyanobacteria. I mean, you may say it's a lot or it's not, but this is the number, but be careful. I mean, it doesn't tell, this is like a modern cyanobacteria. we have no clue what was this percentage a long time ago. Then it's not for all cyanobacteria. There are some cyanobacteria where it's much more frequent. And so, for example, we have now a study of that cyanobacterium more specifically. So it's microcystis, belonging to the microcystis, affiliated to the microcystis genus. So as I was telling you, this is a toxic cyanobacterium. It's blooming in environments where, eutrophic environments. So if you have like higher loads of nitrates or phosphates, they tend to increase the risk for that. And this is one picture illustrating that these blooms. And the problem is that some of these bacteria are producing toxins. And so when this happens, like in drinkable water reservoirs and so on, this is really a major issue. Interestingly, we observed, based on field work on some of these blooms, we observed that carbonate inside the microcystis on some of these blooms. And so again, looking at the genome specifically of microcystis in databases, we could find 300 genomes right now. And you have 1/3 of them contain the CCYA gene, which means really in that genus, the formation of calcium, intracellular calcium carbonate is an important thing. And so you have here this phylogenetic tree, that is representing in a different way. And all the gray shaded areas correspond to the microcystis with intracellular carbonate. We have no clue how this intracellular carbonate affect the biology of this guy. What is the function? Why do they do that? We don't know. And the thing is, since you are producing very high density of these cells, that each of them contain carbonate, you may have really an impact on the geochemical cycle of calcium or carbon in those environments. And that also we have no clue. The second thing, that was again some kind of converging stories, and I think they would not have converged as fast if we hadn't had this bioinformatic approach. On another topic, in Lake Pavin, in the center of France, close to Clermont-Ferrand, we had these regular field trips, and some of our colleagues discovered, again, randomly somehow. Of course, they were interested by bacteria forming intracellular iron oxides, which are magnetites. And by looking at those magnetites, we found that some form intracellular carbonate. The funny thing is that some of these bacteria, they could be micromanipulated. The DNA was extracted and sequenced. And it shows that some of these bacteria belong to this genus, which is called Methylococcus. I had no clue what it was several months ago, but it's possibly, we don't know, but possibly methanotroph. And the funny thing is that Maxime, by running his tool, was showing that several bacteria, numerous bacteria affiliated to Methylococcus contain CCYA, the calcium. Okay, so, and I can assure you that genera in bacteria, there are many. So it looks like not a coincidental thing. Third thing, now we want to really understand the function of this protein, the calcium. Part of it also goes through the determination of the structure of protein, and that's what Geoffroy Gachinian right now is doing in his PhD. So now that we know the sequence of the gene, we can put that gene in another bacterium, Escherichia coli, E. coli, that we know how to culture at high density. So this E. coli is producing a lot of this protein, expressing the gene, producing the protein, and now is extracting the protein and trying to purify it. And the idea is that if you have like a suspension of protein that is pure enough, you may be able to crystallize it and then go to X-rays to determine the structure of this protein. I'll skip that. We can, another way to do that is to, what we can do is that in those bacteria having calcyanine, we can delete the gene. So this is what is called genetics, and try to see what happens when you delete the gene. This is very trivial, the way it says. We have already one PhD student who managed to not delete the gene in those bacteria, but insert the gene in cyanobacteria not forming intracellular carbonate. And she could show that in those mutants were accumulating more calcium than the wild type. But the gray would be the deletion of the gene in cyanobacteria from intracellular carbonate. Try to see, first of all, if they do not form intracellular carbonate anymore, and try to understand how they might use those carbonates. And away from the cyanobacteria forming intracellular carbonate, we can explore more like what we call the dark proteins. So there is still a lot of genomes or the proteins that we don't understand what it is for, what it corresponds for. And that can be the old proteins or parts of the proteins that we don't know how to what they are doing. So this is what Apolline Boulet also has done during a PhD. I'll finish there. Oh, yeah, maybe. So, okay. There are many other topics that now we would like to address in the future. I mean, using this same approach because it's super powerful. One, for example, is that so recently there was the discovery of some bacteria forming silica, and it looks like they might have specific genes involved in silica precipitation. This is very interesting because silica biomineralization. So far, we have thought that silica biomineralization appeared mostly with some algae. You may know of diatoms or sponges, I mean, which are not algae, but okay, in these organisms. And we thought that they appeared like several million years ago, like 500 million years ago. And when it happened, we think that there has been a dramatic decrease of the silica concentration in the ocean. That completely changed dramatically the chemical composition of the oceans. Finding this capability in bacteria raises the question of whether that biomineralization appears maybe in bacteria beforehand, and then trying to understand how this might have been transferred to eukaryotes. So, but the question is what genes are involved, how they are distributed, and so on. So again, we need the kind of approach that I showed you. Okay, I'll just finish, and that will be my conclusion. Sorry for being so long, but 3 points. Okay, then I get, now I'm getting somehow outside of the scientific topic. There are 3 points that I wanted to note and share with you. I think with this work, so I told you that we started started this work. Was it 2015 or something like that? So this has been 7 years. That's it. There are several things that I realized. I mean, maybe it's time, but several things that I realized. First of all, the importance of databases and collections. I mean, I think this work really showed that very well. And to me, somehow they all relate to some kind of idea of the open science. If we don't have that, we can't do that. First of all, the collections. I really realized that having collections of cultures of bacteria is super useful. And instead of getting like 20 different strains, isolating them from every part of the world, and so on, that would take a whole life, I guess. And there you just take the metro and get some of them. There is the same also in the museum, and so and of course many people have worked on them. They have genomes that have been sequenced, so it really speeds up a lot the overall work. Having also the public databases for sequences, protein sequences, DNA sequences. I mean, this has been a long process in biology. And it's pretty common in biology. I mean, you can't publish without depositing your sequences there, but that allows you to do things that you would not be able to do otherwise. And I think for other fields, I mean, that would be a great thing to have. Publications. Okay, they're unrelated topic. Okay? But, just just to show you the story of that paper that we finally got published. Okay, it took time to write it, but I guess, yeah, we had really to be together to write it. And so this is a story. So I guess maybe some other people have experienced this kind of stuff, like submitting to Nature Microbiology, rejection. But that was interesting to see, okay, let's submit it to our open access journal, which will cost 5,000 euros. Then you say, well, no, let's try another one. PNAS, rejection, it takes 1 month, no review. Okay, so let's go to current biology. So it takes 1 day, rejected, so that's better at least. But again, so we suggest you to go to our new journal, iScience. It would be perfect for that journal. And it costs 5,000 euros. So no, we don't go there. So we go to another journal, then it gets again rejected in a few days. And yeah, let's consider publishing it in our sister journal. No, we go to this one. It takes more than a month and it's rejected without a review. So 6 months, 5 different journals, 0 review. That was painful and a bit discouraging, especially because we were so excited to share that. So the first thing I realized that really, by archives, I think at some point are a pretty good option to speed up a bit, speed up the process. Okay, it's not so competitive there, but you know, if you don't want to spend like wait for 6 months before your, oh, it's more than 6 months, right? But wait that your paper will be reviewed and get at least already the data available. That's a good thing. And there are new initiatives like the PCI, which sounds very good idea. The second thing I realized that it was amazing. I mean, all this journal telling me, okay, no, it's not worth for our journal, that for this journal, which costs like 5,000 euros, might be very good. I had just hated that. So I thought, okay, let's follow also some initiative by Montpellier, for example, where they have now their databases of what they call academia-friendly journals. And they've done that in ecology and evolution. So we have started that at the scale of our group. And so you see our group is more like, I don't know, mineralogy, geochemistry, and microbial ecology. And probably this is some initiative we could develop. So trying to understand, you know, where do you pay, you don't pay, you pay for what exactly, and so on. Last, and I'll finish there, nature and success of interdisciplinary. I won't get too much into that, but that's my personal view, I mean, my personal experience. To me, I mean, working at that interface and having those joining or combining those information has really made a dramatic change in the way I see now how I would work biomineralogy, because so far, you know, you could focus on very specific microbes, do very nice things and so on, but you could not grasp the big picture. I mean, what is the representativity of that bacterium, how widespread they are, and so on. Difficulties, for sure. I was kind of a bit lost in the beginning, the first meetings, common language, mind drivers, and so on. Sometimes get recognition for the value of what you do. I mean, it's also, yeah, you are considered as a biologist by a scientist and as a scientist by biologists. Okay, I'm probably in the middle. That reward, I mean, there's definitely no turning back. Okay, so sorry for being so long. I'll stop there. I hope that you got that. Okay, we have really discovered this biomineralization of intracellular carbonate that is widespread in bacteria. We found those genes that are very useful to track those things, and also as a nice way to maybe start thinking about the mechanisms that are involved in this formation of amorphous carbonate. And really for that, we really needed this bioinformatics-mineralogy combination. Thanks, and sorry for being so long.